Welcome, everyone. Thank you for joining us this afternoon. My name is Rachel. I'm part of the care team at PPMD, and I'm really delighted to be joined by the team at Sarepta today as they provide us with some updates regarding their Momentum study, which is a investigational therapy for exon 51 skipping. I'm really looking forward to hearing a little bit more about this program, where they are in terms of data, and what next steps will look like. So we're really glad to have you all here. We will have the ability to dig into some questions and answers after the presentation. So please keep in mind, we do have that chat function in Zoom. You can drop any questions that you have in that chat function. And after the formal presentation, we will go ahead and answer as many of those as possible. So with that, I don't want to waste any more time. I'm really delighted to welcome both Nicole Lamarca from the patient advocacy side, as well as Dr. Fernanda Leal-Pardinas, who is going to share some updates with us. So with that, I'll go ahead and hand things over to Nicole and Fern.
Thank you so much, Rachel, and good afternoon, everyone. We are very happy to have been invited to today's webinar, where we will be discussing the latest advancements in the treatment of Duchenne muscular dystrophy, specifically focusing on our research and findings from the Momentum Part B study. And thank you for joining us as we share this data update. Next slide, please, Nicole. Thank you. We do have a couple of disclosures we want to make. First, if you are a participant in the study, we cannot engage in specific questions related to your participation. So if you have a specific question, just please reach out directly to your study team or to your physician investigator. Today, I will be presenting data that was collected as of November last year, November 2023. In clinical studies, we constantly don't have peeks at the data. We are limited on how much we can see and what we can share. It is important to note that Momentum is an ongoing study, and anything that I share today is what was collected up until November 2023. More data will be shared as the study advances and when the study is complete. In today's presentation, we will be making forward-looking statements about our ongoing research and potential future treatments. These statements are based on current expectations and come with risks and uncertainties. Next slide, please, Nicole. Our goal in Sarepta with Duchenne is unwavering: to develop life-changing precision genetic medicines to one day reach 100% of individuals living with Duchenne muscular dystrophy. Together with Efficacy, the medical community, and those living with Duchenne, we are striving to improve the lives of those affected by this condition through innovative therapies and research. Next slide, please. When we look at our pipeline, it spans from early stage research to marketed therapies, and we're evaluating multiple approaches as we demonstrate our commitment to the DMD community. Our robust DMD portfolio includes various products at different stages of development. Starting with work at the earliest phase, our work in gene editing stands at the animal studies or preclinical phase. Our PPMO technology, which I will be focusing on today, is currently transitioning from early to late stage clinical development or human studies. And we currently have 4 approved products. 3 of them are PMOs aimed at different exon skipping targets in Duchenne muscular dystrophy. One of them is exon Dys51, Vyondys 53, and Amondys 45. And the 4th approved therapy, Elevidys, is the first gene therapy approved for Duchenne in the US. Combined, these approved therapies and investigational therapies underscore our decision and our dedication to addressing the unmet needs of the Duchenne community. Next slide, please. So let me talk a little bit about the PMO mechanism of action. Genes contain instructions that indicate how to create proteins through mechanisms in your own body. Duchenne happens when a specific exon changes in the dystrophin gene, or DMD gene, which is the largest known human gene. This prevents your body from producing dystrophin, a protein that muscles need to work properly. Without dystrophin, muscle cells become damaged and weaken. Now, phosphorodiamidate morpholino oligomers, or PMOs, are designed to bind with precision to target specific exons of dystrophin. Now, imagine this car is a PMO and that an exon map is almost like a road with specially shaped pavers. Now, you can see that when there's a deletion on some exons, the car stops and the connections no longer line up. The car is not able to continue down the road smoothly. If we skip over how the car just did certain exons, we can restore the road in a way that it makes it shorter and useful for the car to pass. This is a simplified way of thinking about exon skipping. By enabling the production of a shortened dystrophin protein, we want to enable the reading of the instructions of the gene more adequately. This backbone chemistry has led to the development of EXONDYS 51, VYONDYS 53, and AMONDYS 45, targeting different exon skipping needs. Next slide, please. Thank you. Now let me talk to you about our peptide phosphorodiamidate morpholino oligomers, or PPMOs. These are similar to the PMOs that I just described, except that with a PPMO, we have affixed a peptide or protein. Here it looks like the oval in purple, which we believe will cause increased tissue penetration and therefore potentially greater dystrophin expression. We hope that this will offer enhanced treatment effectiveness for Duchenne muscular dystrophy. Now, SRP-5051, or vesleteplirsen, is our first PPMO compound, and our preclinical studies show that it was able to reach different types of muscle, including the heart. This was the investigational medicine used in the MOMENTUM study. Next slide, please, Nicole. Let me tell you, I know this is a loaded slide, so let me go and walk through the MOMENTUM Part B study design. MOMENTUM is a two-part global study evaluating SRP-5051 in patients amenable to exon 51 skipping. SRP-5051 is administered once a month via an IV infusion. Now, as I said, it was two parts. Part A, on the left side of the slide, or the darker blue, evaluated several dosing levels to determine a safe and tolerable dose. Part A enrolled 18 patients. We stopped Part A of the study when we identified that an adverse event called hypomagnesemia was experienced by some patients. Hypomagnesemia is when there is not enough magnesium in the blood, and magnesium is important for many body processes, including cardiac and nerve function. It can cause mild symptoms like muscle cramps or more serious health issues like heart rhythm changes or arrhythmias, if not treated. Hypomagnesemia is often treated with dietary changes or supplements. Now, after evaluating this, we designed Part B, which we added daily preventive oral magnesium supplementation. Now, Part B of the study, on the right side of the slide, or the lighter blue, we evaluated dystrophin levels in skeletal muscle at two chosen dose levels, 30 mg per kilo and 20 mg per kilo. Part B enrolled 40 new patients who had not previously received SRP-5051. These patients went the 24-week biopsy, which results we'll be presenting today. Our total Part B safety population includes 22 patients from Part A and previous 5051 studies, that met the entrance criteria, plus 40 participants in Part B, for a total of 62 patients. Throughout Part B of the study, we administered prophylactic magnesium supplementation and/or adjusted the dose of SRP-5051 to manage the hypomagnesemia. Okay, next slide, please. So let's take a closer look at the group of participants who joined the study. As I mentioned, Part B of the study enrolled 40 new patients. These patients were from the age of about 8 to 21 years of age in the United States, Canada, UK, and Europe. Approximately half, so 21 of these patients who participated in the study, were able to walk at the beginning of the study or considered ambulant, and the other half, 19, were non-ambulant. Next slide, please. Now, let me talk a little bit about the endpoints or evaluations of efficacy and safety, as our study is clearly guided by them. I will talk about how efficacy is evaluated in the next slide, but for safety, we examined the adverse events that participants experienced throughout the duration of the study. This study is scheduled to last 5 years, and as I mentioned, it's currently ongoing. Next slide, please. So in this data cut, we evaluate efficacy in 2 different ways. Our primary endpoint is dystrophin production in skeletal muscle by Western blot analyses before and after treatment. Now you can see in the top right of this slide an example of a Western blot. This measures the amount of dystrophin and tells researchers how much total dystrophin is in a muscle sample before and after treatment. In this example, the sample in the center has the most amount of dystrophin. It is important to note that every manufacturer uses a different technique for analyzing Western blot samples, so comparisons should not be made across different companies' programs. Now, the second endpoint, or the second way to measuring efficacy, is the percent of dystrophin-positive fibers, or PDPFs. This is measured by immunofluorescence or fluorescent tagging of the dystrophin in the muscle. PDPF is important because we can distinguish how much of the dystrophin is pure compared to how well it is spread out in the cell, specifically in the muscle cells. With PDPF, we can calculate the percentage of muscle fibers with dystrophin. It gives us a sense of the distribution of the dystrophin of the fibers. And you can see here an example in the bottom right of the slide, 2 samples. And the one on the right, as you can see, lights up more. And therefore, there are many more spread out fibers with dystrophin compared to the one on the left, which has few fibers with dystrophin. These outcome measures in Part B were evaluated in our momentum study at week 28. Now, as a reminder, on the next slides, I will be presenting a snapshot of the safety and efficacy that was taken back in November 2024. The study is ongoing, and this means that the efficacy and safety information will evolve. Therefore, people are still participating in this study, and we are still capturing their experiences on an ongoing basis. Next slide, please, Nicole. Now, let me present our efficacy results. We are pleased to see a significant increase in dystrophin in both treated groups. SRP-5051 has shown a mean dystrophin expression of 5.17% at our high dose. And in our low dose, the expression was 2.8%. We believe that greater production of dystrophin is important in Duchenne, and we hope to show the impact of dystrophin clinically over time. Next slide, please, Nicole. Now, when we analyze the different populations of the ambulatory on the left and non-ambulatory on the right participants in this study, we are encouraged to see consistent dystrophin expression across both groups. To us, this suggests the potential of SRP-5051 in addressing the needs of this diverse population. Next slide, please. Now let's talk about the PDPF results that I just explained, which is the fluorescent tagging or immunofluorescence. We saw an increase on PDPF in both treated groups. On the high dose, SRP-5051 demonstrated a mean of 50.68% dystrophin-positive fibers at week 28. And in the low dose, it was 21.79%. Next slide, please. Now, safety remains a cornerstone of our research. We're committed to closely monitoring and managing many adverse events, ensuring any of the adverse events, ensuring the well-being of our participants. So in the next slide, I'm going to be summarizing the safety events. Thank you, Nicole. As you can see on the top of the slide, it is important to note that mild to moderate hypomagnesemia and/or hypokalemia are very common and are generally asymptomatic, stable, or associated with only mild symptoms. Now, hypomagnesemia and hypokalemia have been controlled with standard supplementation, so that is magnesium or potassium supplementation or changes in diet, and was not associated with any clinically significant adverse events. Now, let me tell you a little bit about the hypomagnesemia. When we started part A of the study, we did not anticipate a need for daily supplementation of the magnesium. The magnesium supplementation and the blood monitoring that participants undergo in this study are because we want to be able to monitor these levels of hypomagnesemia or low magnesium that participants have experienced. Now, we are very thankful for the participants, the experts, and investigators who committed their time and effort to conduct this study and to conduct the monitoring and the supplementation required in this study. Regarding the hypokalemia, hypokalemia is similar to hypomagnesemia, but instead of having low levels of magnesium in the blood, it is low levels of potassium in the blood. Potassium is also important for heart health, muscle contractions, and nerve signals. And both conditions can cause mild symptoms like muscle cramps or more serious issues if not treated. And as I mentioned, both of them are treated with dietary changes in the supplementation. We believe that we see hypokalemia in association with hypomagnesemia because the absorption of magnesium is related to the body's absorption of potassium. When magnesium levels are low, it can cause kidneys to excrete more potassium, leading to hypokalemia. Thus, these two conditions can often occur together and may impact each other. Now, there were serious treatment emergent adverse events in Momentum Part B. We had 4 serious events of hypomagnesemia and 3 serious events of hypokalemia. But as I mentioned, there were no sequelas and there were no clinically significant adverse events. Now, regarding the estimated glomerular filtration rate, or eGFR, that you can see in the line right before the treatment-related SAEs, this eGFR is a basic measure of renal function. It evaluates how well the kidneys are filtering your blood by removing waste and extra water to make urine. We also evaluate thoroughly other measures and molecular biologic markers of renal injury. In this data cut, we've had 4 patients that have shown a decline in eGFR. These declines have been asymptomatic and are not associated with any abnormalities in other biological markers of renal function. When these eGFR declines happen, we discuss internally with our nephrologist and our study doctors and temporarily suspend dosing out of caution. Several of these participants have resumed dosing. We have not determined whether these cases of eGFR declines are associated with SRP-5051 because cases have occurred in older non-ambulatory patients where the cause is uncertain, as some cases may have been caused by background rate or other confounding factors like concomitant medications. So I mentioned in a previous slide, this is a data cut of November 2023. The study is ongoing, and we are continuing to monitor magnesium, potassium, other electrolytes, kidney function, and general safety for this investigational medicine. We appreciate the effort that investigators and participants have given to supplement, monitor, manage, and report their experiences in the study. We could not understand this investigational medicine's potential without your efforts. Next slide, please, Nicole. So let me tell you about next steps. We have reached a pivotal milestone with 5051, achieving our primary endpoint and demonstrating robust dystrophin expression. The safety profile is consistent with our expectations, and the study continues to accrue really important data to further our understanding of the safety and efficacy of this investigational medicine. In terms of next steps, we are seeking a meeting with the FDA during which we will be discussing these results and the next steps for the program. We aim to have this meeting on the third quarter of this year. We anticipate that there are a lot of questions from the community about what to expect next. When will we seek approval? When will there be another study? What are our timelines? Our best answer to any forward-looking program question is that the outcome of this discussion with the FDA will determine what are our next steps. The Momentum study is ongoing, and our tradition is to share updates with the community as more is known. And I do want to say that our commitment to the Duchenne community remains as strong as ever. Next slide. Thank you, PPMD, for your attention, support, and the opportunity to share these results. But we especially want to thank the participants and the families who join clinical studies. The participation and dedication in these families is crucial as we continue our journey to bring new treatments to the Duchenne community. Thank you so much, Rachel.
Thank you so much, Fern. This was a really helpful overview of where you are in the program. And I know that I learned a lot, and I'm sure that everybody listening in also learned quite a bit as well. At this point, we can go ahead and pivot to our question and answer portion of the webinar. So if anybody listening in has questions for Fern, please feel to drop those in the chat, and we'll get through as many of them as we can. I do want to go ahead and start with a couple of questions that were submitted prior to today's webinar about this content. And the first is regarding opening trial sites or seeking regulatory approval outside of the US. Can you provide us with an update on any sort of plans to look at this drug and potential accessibility outside of the United States?
Yeah, so the Momentum study right now is continuing in the US, in Canada, in the UK, and Europe. We have engaged with regulators outside the US and inside the US in the past. And as I mentioned, we're targeting this conversation with the FDA on the third quarter of this year, in which we will share the data with all the that has happened in all the other countries. And that will give us enough information to determine what are the next steps for this program. But definitely, I just want to make it clear that this is an international study. So, yeah.
Excellent. Thank you for that clarification, Fern. The next question is in regard to female manifesting carriers. Are there any plans or any discussions about being able to include female manifesting carriers in clinical trials or potentially in approved therapies?
Yeah. Thank you so much for that question, Rachel. It's actually a really good question. At this time, we do not have any studies planned for female carriers. There is a lot of variety in the experience for carriers and females living with Duchenne, and not a lot of data to support effective studies in this population. We need a degree of confidence from natural history studies when we plan our clinical trials. And since we don't have this, we at this time cannot plan any studies with female carriers. So, yeah.
I think that's a really helpful response, Fern. It's something that so many families desperately want, but it's an area where we still lack so much information, and we hope to learn more and more so that we can do better, truly. As an aside, we actually had this conversation with the FDA in the room earlier this week at PPMD's advocacy conference. So they are hearing us too loud and clear that this is something that's really important to the community, that we be as inclusive as possible. So, pivoting now to some of the information that you shared specific in the slide decks, I noticed
Mhm.
on slide 7 you provided that really nice overview of part A and part B and the different doses. Can you speak to whether anybody in that Part A group who was not being treated prophylactically for hypomagnesemia, did they respond well to treatment and return to baseline when they experienced that drop in magnesium level?
You mean that they went back to the baseline levels of hypomagnesemia, so of magnesium in the blood?
Sorry, back to baseline in terms of did they get rid of all of their symptoms? Did all of their symptoms
Oh,
resolve?
yeah,
Did they recover well?
absolutely. Yes. Yeah. And as I mentioned, of those 18 patients, we give them the opportunity to roll over to the part B of the study. And most of them did. So we were able to follow them, and we're still following them for efficacy and safety. If they would have been symptomatic, or there would have been any issues of serious adverse events related to any electrolytes, we would not have dosed them. They would have been discontinued from the study. Yeah.
Okay, that's really reassuring. And then once you added in that supplementation to help avoid the onset of hypomag or hypo-K, did you see that stop those things from occurring entirely, or did you still see somewhat of a dip, or what did that look like?
In some cases, as you know, there's variability in the response from patient to
Of course.
patient. And the electrolytes depend a lot of as well on the lifestyle that the patients have, their nutrition, their hydration status. So we constantly tell the investigators to remind the families to talk to them about the nutrition and the hydration of the patients, because that's extremely important for electrolyte balance. So when we do see that, we see them sometimes go back to normal levels, or to have really low levels of hypomagnesemia or hypokalemia. And we discuss with the investigators whether or not we will be continue to dose with those low levels, or to pause the infusion. But most of the times they go back to their normal levels, and we're able to continue dosing. And hence, we were able to see a response in our efficacy in our
Excellent.
biopsy.
No, that's incredibly helpful. Somewhat related, I noticed on slide 15 you were talking about that decline in EGFR, and you
Mhm.
mentioned that they paused dosing and then later resumed it. Are you able to speak to how long that pause was and how quickly the EGFR bumped back up?
It depended as well from patient to patient, Rachel. In some cases, the pause was just of a month, so they just suspended a month. And in some cases, it was a little bit longer than that.
Okay.
Yeah.
Okay. And as a reminder to everyone listening in, this is a medication that is dosed monthly. So that means that in some of those cases, they only would have missed one dose and then they were able to then resume.
That is correct.
Perfect. Okay. Excellent. All right. Next up, let me see here. This is a good question about washout. And I think that I already know the answer because you talked about how next steps would be dependent upon conversations with the FDA, but I'll ask it anyway because it's a really great question. And that is during phase 2, one of the qualifications was that there needed to be a 3-month washout if an individual was already being treated with exon 51 or teplersen. Do we know if there would be a washout required to participate in phase 3?
Um, most likely, the reason why we ask washouts in clinical trials in general is because since you're evaluating efficacy, you don't want to have anything that could in some way compromise your results. So when we investigators or when the regulators evaluate the data and say, hey, this patient was in another treatment, how do I know that the effect was related to the treatment that you're investigating or the treatment that they were already on? So that's why we usually ask for that washout period in in the cases of patients that we want to recruit, so that we know that we can control as much as we can the investigational doses that we're giving, and the effect that is going to be seen is due to the investigational drug and not other treatments. So we don't have the final design of the phase 3 study, but to tell you the truth, most likely we will have a washout period. Yeah.
Absolutely. And to your point, Fern, that's something that we see across the landscape in all clinical trials, that there's going to most likely be a washout of either investigational or potentially approved therapies, just to ensure the quality of that data that you all are collecting, so that you have as clean of a data set as possible to then use with conversations with the FDA to hopefully support an approval at some point down the road. Here's a good question about functional outcome measures. So the question is if we have any functional outcome data or results, or is the data that we currently have specific to Western and dystrophin quantification?
Yeah, PDPF. Yeah. So right now we only have those biological endpoints, those surrogate endpoint data. We will have at some point functional data, but as I mentioned, the study is ongoing. So the participants are ongoing those functional assessments. And when we have that information, we will share it with the community.
And unclear timeline on that piece, is that correct?
Yeah, right now
Okay.
everything is, I think the next big step is that FDA meeting that we're hoping
Sure.
to have. And then afterwards, I think that's when we'll have a little bit more information and data to share.
Excellent. So we'll be looking forward to Q3.
Yeah.
So I think that those numbers, those bar graphs, especially that look at dystrophin production, are kind of shocking because they're so good. Um, but I think that there's context for all of these things, and it's helpful to kind of look at it in the larger conversation. So I wonder if you could talk about how those percent increases compare to eteplirsen, or if they compare to eteplirsen. And then I also have a question here seeking clarification from some of the conversation around the percent increase on the investor call. And if you could just kind of clarify what is that increase in dystrophin production, and just kind of clarifying those numbers.
Yeah. So I do want to also give Nicole a chance to answer. So Nicole, do you want to take this question, or you want me to run through it?
Sure. I can start it off a little bit in regards to with the eteplirsen comparison data. I think today we're really here to focus on data from the momentum study, so not making any comparisons between studies or trials. So really just focusing on momentum. And then the second part of the question, can you please restate that for me, Rachel?
Of course. Can you help us understand that increase that we are seeing in those slides? It looks like there's a reference to the investor call here several weeks back, which is incredibly helpful and actually spurred us to ask for, another opportunity to talk about the data because it was so impressive. And it mentions that the data suggested 10% dystrophin production, but the data presented on the slides seemed to not show 10%. So can you just help us understand in very clear terms what that percentage of dystrophin production is? And is that like in comparison to something, or just help us understand those numbers?
Yeah. And Nicole, do you want me to take this one?
Yes, that'd be great.
Sure. So right now we did present an approximate 10% that was a data used for when we did an evaluation of accumulation based out of a modeling exercise. So this was shared when we had our first part A results, and we were estimated that accumulation through time could reach up to 10% of the production. Now, the data that we share today is a larger population. And as I mentioned, Rachel, there's variability in their responses between patient to patient. But we're very encouraged to see the mean production, which is the mean of the of about 5.2%, 5.17% on this larger population. And since it is a mean, there's some patients that have a little bit more, there's some patients that have less, but I think it's still extremely exciting and very encouraging to see this amount of dystrophin in this population. So we still believe that there will be accumulation, but as I mentioned, this is a biopsy and it's a cut through time. So we can only see that 28-week data, and the other thing, it could be done by some modeling exercises, but we haven't done that yet. And but I think it's exciting to see this results. We were certainly pleased when we saw them.
Absolutely. I'm going to ask for just additional clarification on this accumulation-based modeling, because this is, I think, a really helpful concept. So I'm going to restate and make sure that I understand that the 10% was not an actual number that we saw in an individual who was treated, but based on a modeling exercise. So if we're thinking that this is based on modeling, is that with like a hypothetical time point in the future? If it's not 28 weeks, maybe it's 56, or can you just help us understand what modeling looks like for the general population who are not biostatisticians like
so just to clarify it, we may have a patient that has 10% production dystrophin, because all of the data used for modeling exercises is using current data. What we haven't been able to do is a modeling exercise with this current data, with this new data that we have seen. So what is believed is using all of the data that we've had with previous biopsies is you put that into a modeling background, and you're able to more or less evaluate what you think the production will be, and if there's an accumulation. But right now, for this particular data, we haven't done that yet. We're hoping to do it, and definitely expecting to do it, maybe as we plan for that phase 3 call, or even later than that. So.
Okay, perfect. That's really helpful. We've been having a lot of conversations about modeling lately, and so I'm learning a lot myself about what modeling is and does and how it can be helpful for these types of conversations. So I appreciate you digging in a little bit more there. All right. I have another question about how the medication is delivered, and that is it delivered by AAV or a virus, or is a virus involved at all? Can you talk to me a little bit about administration?
Yeah, well, that's a really good question, because it differentiates our ASO or antisense oligonucleotide platform to our gene therapy platform. Our gene therapy uses AAV, RH74, to deliver a shortened, truncated gene. So what we're delivering with gene therapy is a version of the gene, of the dystrophin gene, is just a shortened one. Here, as I showed in the example of the car, the ASO is based out of messenger RNA. And when the messenger RNA is reading it, what it does is that it skips an exon to continue the reading of the DNA, and that messenger RNA will then have the production of the dystrophin, shortened but dystrophin. With AAV, when you're giving the RNA, you have to use a vector because you want it to get to a specific place. Here, we don't need to do that because it's already the RNA messenger. What we added in our PPMO is we added that protein which was that purple oval in that image that we had, that helps even more the introduction and the production of dystrophin. And that's kind of the differences between the two technologies.
Excellent. Thanks, Fern. So to clarify, this is not a viral-based therapy. This is an investigational therapy that is delivered by IV intravenously into a vein once monthly, potentially indefinitely, and is very distinct in that regard from gene therapy, which is viral-based and currently administered as a one-time single dose.
Yeah, exactly.
Okay. Is there any consideration for potentially increasing the dose at this time?
Not right now. All of our doses have been thoroughly studied. That and I just want to give a little bit of background of what I'm thinking we're having this question. I think people might think, well, if there's more, then we will have more dystrophin. But it may not work like that. You know, that we may have a threshold that our cells might just be able to uptake certain amount of drug, and we may be able to see other toxic issues at higher doses. So the doses that we choose for our clinical studies are carefully selected via our robust preclinical studies. It's not that easy to just go ahead and let's just up the ante here and give more dose. And the other important point is that we also had 3 previous studies, which were phase 1 studies prior to this 201, in which we evaluated different dosing levels, and that's also what informed choosing our 20 mg and 10 and 30 mg, sorry, per kilo dosing schema for this particular phase 2. So right now we're not planning to go. We would need to do additional studies, and we don't believe we'll have that benefit or that additional benefit if we go higher.
Absolutely. So what I'm hearing is that you all do a ton of work and look at lots of different dosing levels, both in preclinical and early stage clinical work, to understand what that ideal dose is in terms of safety and efficacy, and making sure that we find that happy medium between the two. Is that fair?
That's fair.
Okay. All right. I think we've touched most of the high points as I look at the different questions here in the chat. And so I want to just extend my gratitude to both of you, Fern and Nicole, for getting on, sharing these updates, answering all of my questions. I think it's been really helpful, but I do want to give you each a chance to have the last word before we go ahead and wrap things up this afternoon.
Sure.
Nicole, do
So
you
I
want
can
to
go
start?
first. Yes, absolutely. Thank you so much. I just wanted to let everyone know, if you have any questions, please feel free to reach out to Patient Affairs at Sarepta. So it's advocacy at sarepta.com, and we are happy to answer any questions that you may have if you felt that you thought of something after the webinar today. So please feel free to do that.
And I would like to say two things. The first one, if you still have questions specifically about your patient, reach out to your treating physician, reach out to the study team. They should have the information, and if they want more data, they will reach out to us. We have constant communication with them. We talk to them almost every week. We certainly email with them every week. So talk to them, because we are limited but how we can discuss, but you are not limited of how much you can discuss with your treating physician. So reach out to them. And secondly, just say thank you again, because there's no way we can understand this without the participation of the families, the patients, and the communication and commitment to the DMD and their kids. So we really, really appreciate all of the support and your contributions to science and to understand better how this is working. So thank you.
Thank you, Fern. Thank you, Nicole. As a reminder, this is recorded, so this will be available on our YouTube channel, on our website, so you can go back, take another listen. If you have other questions, as Nicole shared, you can shoot the Sarepta team an email. You can also reach out to us at PPMD. You can shoot me a note, or you can reach out to the care team at parentprojectmd.org. We're always happy to connect and point you in the right direction so that you can find answers to your questions. I want to extend a quick thank you to both Fern and Nicole and the rest of the Sarepta team for joining us today, as well as a thank you to all of you for tuning in and listening. We hope that you found this helpful. So with that, we will go ahead and conclude today's webinar recording, and thanks for joining us. Have a great afternoon, everyone.